And he got his bachelor's degree in biology from Whitman College in 1977. And after that, he got his master's degree in microbiology from Northern Arizona University and his PhD degree in microbiology from Virginia Tech in 1986. And after that, he did 2 years postdoc at Idaho National Lab. So he moved to Oregon State University in 2016. And his research involves exploring the extent, the diversity, and activity of the subsurface biosphere in continental and seafloor settings. And he also studied the microbes in the subseafloor sediments and crustal basalts. The microbial enhance the calcite precipitation and its application in contaminant remediation and as a geoengineering technique. He also studied the biogeochemical cycling of carbon. So he led the decade-long census of deep life, a global survey of microbial diversity in the deep subsurface. And he has led or participated in 18 scientific drilling or sampling expeditions. And he has also co-authored over 95 peer-reviewed publications. And with that, I will let Dr. Colwell to start.
Okay, thank you, Kehua. I really appreciate the chance to visit with all of you. Would love to be there in person, but really appreciate the fact that I'm able to present to you, even though I'm not there in person. And I would say first, just an attribution to where I work at Oregon State University and in the town of Corvallis, which is located in the traditional homelands of the Marys River or Apinafew band of the Kalapuya tribe, which was after the treaty in the Willamette Valley in 1855. That tribe was forcibly removed to reservations in Western Oregon where they live today, or where descendants of them live today, along with or on what's called the Confederated Tribes of the Grand Ronde community. So the work that we do here is on their land, and I want to make that known. So the I guess I would like to start by thanking Kehua for this opportunity and saying that I remember meeting her the first time at a Gordon conference that perhaps Peter had organized in Corpus Christi, and appreciated the presentation that she gave, and thought that since that time have enjoyed the collaborations that we've tried to get started and sustain. So I'm really grateful for that. And so I guess when I begin, I always think, okay, how did I get to this point where, as a microbiologist, I was curious about the deep subsurface? I mean, I was always interested in general in life in Earth environments, and so the fact that I started studying Earth microbiology or geomicrobiology isn't too surprising to me, I guess I would say. But the fact that I started looking in deep systems, maybe it's not so clear. So a little bit of background on that. So I worked for a number of years after graduate work at the Idaho National Laboratory in eastern Idaho, and you can see in this map in the upper left-hand corner where Idaho is, of course, and then the Idaho National Lab is about the size of the island of Maui, and it exists in this desert in the northern part of the Basin and Range Province, and it was created in the 1950s as a place where they could conduct research studying various types of nuclear energy. Well, as you probably all know, over a number of years, doing that type of research, there's considerable waste created. And that happened not just in Idaho, but of course, in many places in the world, many places in the United States, where the Department of Energy, at least in the US, where the DOE became responsible for the waste that was created. So I was drawn into this interesting opportunity to work in not just amongst microbiologists, but amongst geochemists, geophysicists, geologists, hydrologists, to try to resolve problems related to waste in Earth environments. And it really took people from all of these different areas to sort of study this and get to the point where we were able to resolve some of the problems. They're not all resolved. But I think, where I first learned, I guess I would say, would be studying a fractured rock aquifer that is quite deep. Well, not super deep, but it's 65 meters down in rock, where wastes were injected in eastern Idaho. And again, this characterizes what has happened elsewhere in the US. And a contaminant plume of TCE, chlorinated solvent, was then introduced into that otherwise very pristine aquifer. And over a number of years, what we discovered was that organisms that naturally occur in this aquifer that cycle methane, some of which make methane as a waste product, so-called methanogens, and others that consume methane, so-called methanotrophs, they're both present in that system. And the methanotrophs, as it turns out, are able to, whether they like it or not, they happen to be degrading the trichloroethylene gradually in that location. And so I guess what I would say is I drifted into this area almost out of necessity. That is, there was an opportunity to conduct research, and oh my gosh, it ended up meaning that I worked with people from a number of different disciplines, not just microbiologists trying to figure out a microbiology problem, but a complex Earth science or Earth system science problem. And it became very exciting. And I think the result was that we began to develop technologies. They were not fully developed at that point, but because of the need to clean up these sites and understand more about them, we began to learn how to use relatively large drill rigs to collect core samples. And this, of course, was already done. The geologists knew how to do this, but for us, the trick was to figure out how to do this without contaminating the core sample with microorganisms from drilling fluids or even from the surface. And we learned right away that the microbial communities, as we'll talk about, are really quite low, quite minimal in these environments. And so we had to develop strategies, tracers, for instance, to distinguish between what was introduced from the surface or from the drilling fluid and what was pristine or indigenous to that environment. And so I show here that we sort of were doing this work on land. It wasn't long before drill ships like the Joides Resolution here began to search for microbial communities in a long-running geology-driven program, but microbial communities in rocks and sediments of the seafloor. And then we also, in some cases, explored deep mines. For instance, the deep mines in South Africa, which have been studied. Of course, they produce gold, but studied extensively now for the biogeochemistry, the microbiology that exists there. And there are a whole series then of technologies, some of them probably that started in the medical field, but that were, you could say, co-opted or used now by environmental microbiologists to understand what we can about life in the subsurface. Okay, so that gives you a little bit of a background on how I got involved in this. I guess I would say or confess to having a long-term love or appreciation of geology and geologists. I was blown over by this as an early student, even thought about changing my major at the last moment. So So it's not unusual when I think about myself ending up in a field that would be called geomicrobiology. Okay, so what I'm going to talk about today really is, in a general sense, the life that exists in deep subsurface environments, and probably come at it from a couple of different perspectives that would include understanding how many organisms are present there, how they may be distributed, the different types of organisms, and their activities or their relative activities. And then I'll talk a little more specifically about environments that I've studied a fair amount, some in conjunction with Kehua and Peter and people at the University of Texas, in marine sediments where methane is produced. One of the first things that microbiologists want to know when they study a new environment is how many cells are present, what is the microbial abundance. And even though this isn't one of the early studies, those first started in marine sediments in the 30s, actually. But this figure, published by Kallmeyer in 2012, shows what we understand of cell numbers, shown as log cell concentration along the x-axis, and log depth in meters below seafloor on the y-axis. And what you can see for these different types of environments, a general trend, right, that the numbers of cells start off pretty high in many cases. This is in the billions of cells per cubic centimeter near the surface of the sediments, but then they decrease. They all show these trends of decrease. But even if you get down a kilometer into the sediments, for these ocean drilling program, the black symbols, which are mostly collected near the continental margins, even if you're down a kilometer, there's still 1 million cells per cubic centimeter, far less than you would find in the surface or in surface soils, for instance, but still a respectable number of microbial cells. And if you go to other places like the South Pacific Gyre, which is, a desert, oceanographically speaking, very little productivity in the overlying water, you still have a fair number of cells in the superficial sediments, but as you go down into these sediments, they decline, but at a much steeper rate than if you are in these coastal margin sediments. Perhaps makes sense in many ways. And so there's a correlation to the mean sedimentation rate and the distance from land or ocean productivity here. And so we understand actually a fair amount about microorganisms in, not everything, but in marine sediments. It's a little bit harder when we start talking about non-sedimentary crustal systems, and certainly the continental subsurface contains some mysteries still, as do places like fractures beneath the Antarctic continent, which we're not going to talk about, but these are places that really haven't been explored. Okay, so if we go into marine sediments, and still we're talking about the numbers of cells and their survival, as we go down in sediments, and this is a rather short section, only down as deep as 7 meters, and this is, I believe, in Denmark, seafloor sediment in Denmark. You see a chemical profile that's familiar to geochemists in marine sediments and microbiologists. So where there's virtually no methane near the top, and there's a concentration of methane begins to increase at some depth. And in a sense, that's at the expense of sulfate that is high near the seawater and decreases. We'll look at this more later. This is referred to as a sulfate methane transition zone. And geochemists understood this for a long time. Microbiologists sort of refused to appreciate the significance of this and that microbes were responsible until microbiologists found those microbes and identified them and began to study them. And then they said, yeah, the chemists are correct. There is a microbial process here that's drawing down both sulfate and methane. So in this location, the SMT is about 3 meters down. And I think what I really want to show you are these curves on the side, which show cell numbers per gram of sediment which are quite high, again, near the surface, but they drop down 2 orders of magnitude by the time you're 7 meters into the sediments. And if you look at this other property, cell generation time, this is the period of time that it takes for cells to divide. So near the surface, where you could think of cells being quite healthy, they have that rather short generation time. So maybe one cell will divide to be 2 in about a month or so. Okay, but as you bury these samples, the microbial communities, in a sense, that, well, they reflect longer generation times at this point, beginning to reflect something closer to decades before they can divide. So fewer cells with increasing depth, and those that are present are becoming constrained. And from the same paper published by Starnowski a few years ago, they looked at other things, and this is the number of OTUs. This is operational taxonomic units, which is a way that microbiologists say species of microorganisms. So in this case, you see similarly through these different zones or depths, a decrease in the number of species. And the number of cumulative generations, so it begins to increase. So you're accumulating new generations of organisms, and it's increasing at some rate in the shallowest sediments. But then as you get into these deeper sediments, the rate of cumulative generations begins to drop off. So you have, in a sense, fewer and fewer generations to build upon. So the implication for this is that a rate of evolution, possibly, which is, we typically think of evolution as being dependent upon growth, that is, cells need to divide to introduce an evolutionary component to their genomics. This decreases. And the surviving cells that are down at this depth, and certainly below that, carry what remaining genetic information they can have, or they have, from this this sort of reduced number of species that are dividing less and less. So it's not, I mean, there is a biosphere down there, but it's not what we commonly think of near surface environments. It's much harsher than that. Okay, so I have a couple slides here that that I use to introduce the concept of molecular signatures or molecular biology as a way to study microbial diversity. And it turns out you can use these signatures to study more than just microbial diversity, but microbial function and activity. All of these things are possible, but it's actually very much a revolution that has happened over the last few decades in terms of how we view microorganisms. For many years, microbiologists tried to grow organisms, and you can only grow a small number. And so of those that exist in nature, we figured out how to grow the ones that are most important to us, those that cause disease, for instance. However, a vast majority of organisms we cannot grow. And so what microbiologists, along with biochemists, figured out how to do is take a microbial cell depicted here, and these are size scales, so you can see human hair for comparison and a range of thickness, and then scale that down, and you've got a microbial cell that a healthy one might be 1 by 5 micrometers in size. And all these little green dots within that cell are referred to as ribosomes. This is the protein-making machinery or micromachine that exists within not only microbial cells, but all living cells. And so a cell, in order to be alive, has to have a ribosome, at least as we understand it here on Earth. So microbiologists and biochemists looked at the ribosome and said, well, if this is essential to life as we know it, to living cells, let's look at the chemistry of this essential unit and study that in detail. And that's shown here, a fragment of ribosomal RNA, which was just the first gene, or the gene that codes for ribosomal RNA, was simply one of the first genes to be looked at intensively by microbiologists. In a sense, they selected it because it's so required in life as we know it. They selected this gene to use as a point of comparison, with the basic idea that the chemistry of this gene, the base pairs by which it is constructed, would be identical for species that are the same. However, as you get to more distant related representatives of a group, there would be slight variations, and that's shown by these letter exchanges in a sense. You might be able to exchange uracil for a cytosine, that sort of thing. And this could then be detected originally in a very laborious chemical manner and teased out into family trees that would show relationships. And perhaps you can see the similarities in the dark bands and the light bands here, and this somewhat more distantly related organism. So this is the basis for molecular biology and how microbiologists, and now all biologists, have made comparisons of all life, essentially. And this is, now many people would probably tease me and say, well, it's 5 years old. There are many more recent trees, but I do like this one published by Hug a few years ago that looks at the 3 domains of life: eukaryotes, which we're familiar with because we exist within them, way down here in this portion of the tree; archaea, which are actually single cell, simple organisms that are very related to eukaryotes, as it turns out. They don't look like us, but they are related to us. And a chemical and a genetic relationship, they're very similar. And bacteria, this other domain, bacteria. And I think a main point that I want to make here again is that many of these are uncultured. All the red dots you see here, we see evidence of these organisms in earth samples or in the environment. However, we haven't been able to grow them in the laboratory. So we have some limited knowledge of their activities and so on. There's a large evolutionary divergence in this uncultured fraction. So all these red dots spread across this large tree of life in a sense. And so we know that there's a lot of evolutionary divergence there. Many of these uncultivated groups are from Earth environments, and that's simply because they haven't been studied enough. Okay, so one of the things that we can discover beyond just the relationships are the functions of these cells. If we're able to get enough DNA, then we can sequence not just the genes that relate to how we identify or classify the organisms, but we can identify the various functions that these cells may have in their environment. And this is sort of a great example of an organism that was captured back in the early 2000s. And then it was described first using what's referred to as a metagenome, where they sequenced the entire genome of this organism. It came from a fracture almost 3 kilometers deep in a South African gold mine. This is where it was originally found. It has a thermophilic lifestyle. That is, it's hot down there, and this is what this organism is adapted to. And even though it's a bacterium, as I showed on that early slide, it has archaeal genes. That is, it's received genes from archaea that allow it to fix nitrogen and fix carbon from its environment. It happened to be a dominant member of that community, so almost 99% of that community, and that's why they were able to assemble this genome so successfully early on. And it uses hydrogen and carbon monoxide as its source of energy. That's apparent from its functions. So it's capable of an independent existence in these deep gold mines, but subsequently, similar phylotypes have been detected in seafloor sediments. So it's not just confined to the gold mines. That's just where it was found first. And now they've found it in many places. And only this past year has it been domesticated, as we would say. So what do I mean by that? You can grow it in the lab now. A group of Russian scientists have determined how to grow it in the lab. But we knew before we could grow it, we knew it was there, and we knew it had capabilities like the ability to form endospores, a resistance stage, or to resist stress that would come from oxygen radicals like peroxide or superoxide. And we also understood these other properties of the organism. So it was really an intriguing organism we knew a lot about. It was sort of a poster child in a sense, but we were unable to grow it. So the whole effort to understand life in the subsurface frequently involves an understanding of the diversity of organisms. And in 2010, I became a part of a program that was sponsored by the Sloan Foundation, part of the Deep Carbon Observatory, which was aimed at studying carbon in deep subsurface environments on the planet. But a component of that was called the Deep Life Community, and we created the Census of Deep Life. And the basic idea here was to try to understand more completely the role of life in deep carbon cycling, possibly the diversity of organisms, the biogeography of these organisms, look in both terrestrial and seafloor settings, a number of different geologic places, and study primarily bacteria and archaea that exist in these environments. And this is only possible because of the high throughput sequencing of 16S genes and subsequently metagenomes, so the whole genome of collective microbial communities that were sampled. So this was a community effort where essentially we put calls out for people around the world to send us DNA that they had extracted from subsurface environments. And towards the end, this is a map that we created of the different places in the terrestrial subsurface shown here with the white dots, or the marine subsurface shown with the black dots, where we collected samples, or where samples were sent to us. This was sent to the Marine Biological Laboratory at Woods Hole, where Mitch Sogan headed up the high throughput sequencing. And then we would return the sequence data to the individual investigators. And in a sense, this was in exchange for building a database of what we understand about these deep environments and the microbial communities there. We're in the process of, so all these individual investigators, many of them published individual papers on their particular setting, but what we're trying to do now is assemble a picture of the microbiogeography of the entire subsurface based on this set of samples that had been contributed over about 8 years to the Census of Deep Life. And I'm going to show just a couple slides here that represent some early findings. Emil Ruff is leading this, also of the Marine Biological Laboratory, but there's a whole team of us who are helping Emil, but he's really the head of this effort. And what we see here are two plots that show on the y-axis observed ASV. This is like species. So I used the term OTU before, but this is another term for species. So we're talking about as many as 600 species here for archaea and for bacteria. And you can see the scale changes for bacteria. Typically, in most environments, you have many more bacteria that are encountered than archaea. And it's classified according to this, the blue side, which are a set of marine samples, and the terrestrial side, which is gold. And what you can see here is that these estimates are based on large numbers of samples. So 70 archaeal samples from marine settings, 182 from terrestrial locations. And each one of the dots represents a single data set. That is one sample that was sent to the Census of Deep Life and that was sequenced for its microbial community characteristics. And so within that dot or each one of these dots are numerous species that represent that. So if we're just looking at the number of observed species, we see some interesting relationships here. For instance, that archaeal richness is higher in marine systems than in terrestrial systems. So in most other environments, you see that, well, I guess what I would say is this appears to indicate that the marine sediments or marine rocks are a domain where archaea prefer to exist, and they may even dominate over bacteria in terms of numbers of species that are represented. In this depiction, this on the right side, this is bacteria, and this shows that in marine and terrestrial environments, the number of species is more equivalent between these two systems, even though overall the number is higher for bacteria, the number of species is higher. It's more or less equivalent between the marine and the terrestrial environment. And if we do non-dimensional, non-metric, multi-dimensional scaling analysis of this data, which is one of the only ways that we can actually look at this, because each one of these dots, which represents, again, an individual sample that has numerous species within it, each one of these dots, it would be hard to see it in species space. And so using an NMDS plot, we can reduce these dots to essentially 2 dimensions. And what we see here is that in these 2 dimensions that most completely describe the distribution of these in this NMDS space, we can see that marine communities and terrestrial communities are distinct from each other. So not only might they differ in terms of number of species of a given group, but the types of organisms that are present are different. And that means essentially that for both archaea and bacteria, we have different types of organisms in these different settings. So, you know, it's possibly not too surprising that this is the case. You know, the 2 environments are quite different. Marine sediments from a terrestrial landscape where you may have freshwater percolating through, but this is really a comprehensive study that demonstrates that it is apparently true that the systems differ markedly. Okay, so one of the things that we often talk about related to microbes in the subsurface, or it's a next level question, is, well, how active are they? And so I want to talk about that now in a sense. And I use this slide to talk about microbial activity in the subsurface as a way of comparing it to the surface world. If we look at one scale, which is carbon dioxide production, which might be a measure of microbial activity, how actively they produce carbon dioxide as a waste product through respiration, for instance, or on this lower axis, we've talked about doubling times. So in the past, well, what this would say is a doubling time of 10 to the third seconds, 1,000 seconds. I mean, this puts it in the very rapid doubling time, but you can see the scale here extends down to 10 to the 13th seconds for a doubling time, meaning how many seconds are required for a cell to divide and make 2 cells. And what you can see here is that the surface world that we're familiar with, where we may have fermenters that are making food, for instance, or handling the waste that we produce, sludge digesters, these systems are engineered to operate at very high activity. So you have high production of carbon dioxide or a very rapid doubling time. And some natural environments are on that same scale. So for instance, surface soils, maybe lake sediments, that as you get into the deep subsurface, possibly deep sea waters or subsurface aquifers, and definitely down into deep rock formations or arid vadose zones, you tend then into systems where the organisms are more in a survival state, possibly even in a state of preservation or suspended animation in a sense, simply not doing much down there. And so this, I think, shows the scale or the range of activities that we're familiar with in a broad sense in terms of carbon dioxide. And then looking at it in a different way, if we look at metabolic rate in terms of moles of electrons per year, per liter of material, might be water, might be a liter of earth, for instance, then what's shown here by the different colored bars is a whole range of different metabolic processes that are carried out by microorganisms. So hydrogen oxidation, for instance, sulfate reduction, methane oxidation that may be anaerobic or aerobic. And what you can see here is that these metabolic rates span many different, many orders of magnitude. Some of them might be from the very surface type environments where the activities are still quite high, but others might be coming from very deep environments. And so, for instance, if we just pick methanogenesis, this olive green colored bar, you can see some rates are really quite low, but they range upwards so that there can be environments where, earth environments where methanogenesis is happening quite rapidly. So it just, I think, gives an appreciation of the range of activities, although many of these deeper subsurface settings, the activities are really quite low. Okay, and we get at this a little bit more through this paper that was published last year by James Bradley and his collaborators. This is a terrific paper that looks at the power relationship or the power utilization in subsurface environments and how microbial cells are oriented towards using power, which is a way of thinking of their metabolic activity. And so this looks at a volumetric distribution and flux of sediment, cells, organic carbon, and energy across major marine sediment catabolic zones meaning zones of metabolic activity. And this is, I should make it clear that this is a global consideration, but it's the quaternary subsea floor sediments. So not super deep, what you might consider as still more active portions of the shallow sedimentary subsurface. And it is, again, it's sediments. It doesn't say much or anything really about rocky environments or the continents, but this is a nice way of looking at this. And one thing that you can look at, here's the volume of these environments varies considerably, and they're color-coded. So the green represents oxic environments. The purple represents sulfate reducing environments where organisms that reduce sulfate as a electron acceptor exist. And then the methanogenic environments are colored orange here. And so you can see volumetrically, there's a very large area that is inhabited by the methanogenic zone, or as characterized as a methanogenic zone. And then a much smaller area, very small, where it's actually oxic. And this is just the surface sediments. And similarly, cell abundance, because probably there's such a high volume here, cell abundance is much higher in this methanogenic zone. So even in these quaternary sediments, methanogens are really quite, or the methanogenic zone is quite important. But you can see this relationship sort of switches when you look at the power that's used by the organisms in these environments. So even though cell abundance and the volume is quite low, there's a huge amount of power that is used by cells in this upper oxic zone. And this is shown here in this figure, again, reflecting cell abundance on the y-axis and power. This is power per cell in watts. So even though these numbers are quite low, this is the high end of the scale, colored green, where on a per cell basis, there's a fair amount of power that these organisms are going through. And Bradley, I think, nicely translated this through to plots of the planet, the seafloor planet, the planetary features on the seafloor, so that he can plot here on a global scale and depth integrated for sediment layers, the power per cell in watts again. And this looks at it in three different views, whether it's the oxic, more surface located areas, through the sulfate reducing and the methanogenic. And this scale down at the bottom shows where the power per cell is highest, where it's bright red, and lowest where it's blue. And you can see how this differs depending on where you are on the planet. And of course, I would say also that he's working with limited number of samples, but of course, extrapolating from what we know from these studies on how much power is consumed. And you can see that methanogens, for instance, these organisms that produce methane, their domain where they're important in these sediments is really on these margins. So where there is no color, that means there's an absence of that corresponding catabolic zone. So in all these areas, methanogens are not present in the quaternary sediments. They're likely present in deeper sediments, possibly. Okay, and just I want to say briefly that there are a number of processes that we really don't understand much at all about yet. They have been proposed or hypothesized, and they're intriguing, right? That sediments that contain radioisotopes, for instance, and the decay associated with those isotopes can split water and create reactants and oxidants that microbial communities in the subsurface can thrive on. This, to me, is an amazing sort of departure from many of the strategies that we understand for microbial survival, but this one is now thought of as a way of supplying organisms in, for instance, the South Pacific Gyre with their energy and their oxidants. And in other cases, you might have halogenated organics that serve as oxidants for dehalorespiration, another peculiar possible way of surviving in these environments. But really, we're still in the process of understanding. And on the left-hand axis is what we would consider a more classical redox sequence that describes microbial metabolisms in these environments. Okay, so in the last couple minutes that I have, I'd like to talk about methane hydrates, seeps, and the microbes that exist there. This is an old map that shows, now pushing 20 years old or getting close to that shows a distribution of these unique formations, primarily along continental margins, where you have methane in high concentrations, sometimes seeping out, and the associated microorganisms. And in some cases, this peculiar material hydrate, a mineral form of methane and water. And so this figure, published a number of years ago out of Anchi Boetius's lab, depicts this schematic where you have a large source of methane coming from below, formation of hydrates under the proper temperature and pressure conditions, and then, in a sense, a biofilter near the surface or above where the hydrates exist, where the methane that may be released from the hydrates is consumed by microbial communities. These can be aerobic methanotrophs if they exist in the overlying water, but in most cases, these are anaerobic methane oxidizers that exist along the sulfate methane transition zone, or the SMT, as we referred to in the very beginning. But this methane filter really keeps a large amount of methane that exists in the subsurface from entering the hydrosphere, so that methane emission into the hydrosphere is quite low. However, the rates at which this anaerobic oxidation of methane occur can range over many orders of magnitude. So it can be quite fast in places like the Black Sea, in microbial reefs, but then in deep lake sediments, it can be quite slow. This is the actual reaction where sulfate from the overlying water, along with methane, is anaerobically oxidized. The methane is anaerobically oxidized, and you have, as a result, sulfide and bicarbonate plus water. And these are photomicrographs of the organisms that are responsible for this. Now, we studied this in particular in several environments where you actually find a peculiar pink biofilm in fractures that exist in the subsurface. So people were discovering these, and this ended up being a new way that we saw or described a way that microorganisms would respond to the presence of or the movement of methane through these systems. So whether it was diffusion dominated or seep dominated, microbes are involved. But in this case, we saw this pink slime, which you might be able to see down at the bottom left on the end of this spatula. So I would just say that when microbiologists see something like this, it startles them, because usually you cannot see what you're studying. And so when you see pink goo and you see it repeatedly in marine sediments, it's notable. And so we found these in fractures, and so we believe that this is present primarily in fracture-dominated systems. But what we went on to study is how microbial communities respond in environments where there are different rates of methane flux. This is from a paper that Weili Hong published in 2016 that shows schematically where if you're near for instance, a seafloor mud volcano where there's a high rate of methane flux, and you depart from that, so move away from it, where the methane flux is much lower, sort of off axis of this sort of feature, then you have high rates of anaerobic methane oxidation occurring near this high fluxing methane, but then where methane is still being produced, you have lower rates, and essentially different ways of understanding how these communities are coping with methane that is moving through the system. You can have an aqueous pump sort of setting where physical processes are so strong that you're having cycling of sulfate into the system, so high rates of AOM, or anaerobic oxidation of methane, occurring here. But at the opposite end, AOM is much lower, probably takes on the order of centuries or millennia to build up things like authigenic carbonates. And what we did most recently, and this was led by Weili Hong and Scott Classic, was to look at environments that are considered to be in steady state, that is, where there's no gas seepage. And if you look at the chemistry, sulfate decreases on a linear axis and methane increases below that in a linear fashion. And so you would say that the anaerobic methane oxidation is happening here at the sulfate methane transition zone. So this is a steady state system. And to contrast that with non-steady state systems where you see a different pattern in the sulfate diffusing into the sediments, and also in the methane as it's being consumed, or in a place where you have methane seepage. And what we wanted to do was connect the modeling that Weili had done, the reactive transport modeling, to describe over centuries how methane is being released from these different systems, to a characterization of the microbial communities. And I think one of the most notable things that Scott picked up in this is that that if you were looking at diversity, a measure of the different types of microbes present, that in these no gas seepage locations under steady state conditions, you do not see a decrease in diversity across the zone where there are high rates, where AOM peaks, that is where you can detect AOM, even though it may be low. You do not see changes in diversity in the microbial communities. However, in these more active locations, non-steady state or methane seep locations, if you look at this measure of diversity that biologists use, you can see that there's a decrease in the diversity of samples as they approach and go below the zone of anaerobic methane oxidation. So what we're seeing here is a signature conveyed by the molecular biology, the biodiversity of the system, that reflects what's happening in terms of the dynamics of methane fluxing through the system. And I think I want to finish up with a slide that describes, in my mind, but I'd love to talk to people about this, how gradients in subsurface environments or geologic settings in general determine where microorganisms may survive and how they survive. And this is rather arbitrarily pictured on an axis that goes between chemical features that organisms experience or physical properties that they experience in their environments. But what I've shown here are natural processes in black. And many of these things we would be familiar with, that we're familiar with in geologic settings, or human-induced processes where we may be prospecting for some type of fluid of value, or perhaps creating a repository in a system, or maybe even testing things in the ground. All of these things are processes that enhance or change microbial activity in subsurface systems. And so I think originally it was a set of natural processes, but now superimposed on that, we have a number of human-induced processes that we can study as well. So I just want to wrap up with a couple slides here to remind you that subsurface life, and this is really below the horizon, soil horizons, or below the shallowest sediments in the seafloor, it is abundant, but it does tend to diminish with depth. And yet you can go quite deeply and still find cells. These communities are diverse, and they differ depending on where you are. And there's still quite a bit that is undiscovered yet in terms of their functionality or what they're capable of. In general, these organisms are metabolically quite slow, and they end up being tuned, we think, to very long-term survival in these environments. And we still don't fully understand all of the redox processes that they might use in order to survive in these environments. And we're just getting to the point where we can merge genomics and molecular data into reactive transport models that can provide insight into what the crucial subsurface processes are, maybe those processes that most interact with surface environments like carbon cycling, for instance. And one final slide where I'd like to thank the several colleagues that I have, some of them students, many of them students at Oregon State, that I've had the good fortune to work with, but also people from elsewhere that have helped, to explore some of these environments, these deep environments that we're continuing to look at, as well as the sources for funding for this work. The Sloan Foundation, the Deep Carbon Observatory, IODP, NSF, and the Department of Energy. And so I would finish up there and be happy to answer any questions if people have any.
Thanks, Rick, for the interesting talk. So I think we have 4 minutes for questions. So feel free to unmute yourself if you have any questions. Any questions? Oh,
Looks like Fritz has a question.
Yes, could you hazard a guess about subsurface Mars life?
Yeah. So I left out the slides with, space stuff, which I also really like thinking about. I think that, so NASA, is very careful about saying, we have discovered or we're going to discover life. And they talk about looking for signs of ancient life, that sort of thing. And what I, as a microbiologist, reading periodically, I've seen, for instance, that there's some discussion about geophysical demonstration of liquid water in the subsurface. I think if there is actually liquid water in the subsurface of Mars, possibly saline water, I think that would be the best place to try to locate life right now that might exist on Mars. Wouldn't surprise me at all if it did once exist on a watery Mars. So I think it's quite possible.
Thank you.
Yeah.
Yeah, I have a quick question. At least I hope it's quick. What's the sort of limit to this ability to wait around for the right conditions, do we think, in the really deepest? I mean, we discovered thermophilic bacteria in the Chicxulub impact breccias, that 66 million years later. I mean, are those, are they just kind of waiting around for, resurgence of the right condition, or are they just barely hanging on, as a evolutionary strategy?
Yeah, I don't think that microbiologists have come to terms fully with it. I think if you approach it from a chemical perspective, there are organic chemists that will say, well, DNA has a certain period of time that it can last. And even if you protect it in these ways, it will be exposed or it will be oxidized at a certain point, and you'll lose the capability to, transfer genetic information. But then I think actually some of the findings from places where they've looked in fluid inclusions of very old rock, I think is it the Salida formation in New Mexico and maybe West Texas. It's a salt formation. It has fluid inclusions, and a group from Penn State had found microbes in those fluid inclusions and understand the geology of the system enough to say, well, the last time that material was near the surface was, I think, 250 million years ago. So we don't know what has happened in the intervening time, but those fluids are sort of entrained geologically. So obviously they're hard experiments to perform to test this, or hard to convince anybody that it works. But the best thing that can be done is to say, well, we know these are the ages of these sediments. There are microbes there. They may not be the original microbe, but there's still this slow rate of turnover. And so, for instance, the organism that we may see today may be using pieces that were left over from an organism that was buried a long time ago, and those pieces still remain and can be reconstructed by an existing organism, however slowly, into something that we would now recognize as a viable cell.
Very interesting. Thanks.
Sure. Thanks, Sean.
okay. Thanks, Rick, again for your interesting talk. And so if you have more questions and want to talk with Rick, we still have slots available in the sign up sheet. Thanks. Thank you.
Thank you all for coming. Thanks, Kehua.
Thank you.